the demo video for dimensionality reduction assignment, and I'll be going over the first collab. And for this section right here, we are just setting up our environment and importing all the libraries and stuff, and various dimensionality reduction tools like PCA, UMAP, and TSNE, and then others. These libraries will mostly allow us to pre-process the data and then reduce its dimensionality and visualize the results effectively. So here we are loading and exploring the image dataset. We use Olivetti faces using fetch Olivetti faces. Um, this dataset contains around more than 400 grayscale images of 64 by 64 pixels flattened into 1D arrays for processing, and then we also visualize a few sample images to understand the dataset we are working with. And then right here we are starting with the dimensionality reduction techniques, and the first one is locally linear embedding. So for locally linear embedding, in this block we are using LLE to reduce the dimensionality of the dataset to 2D. LLE preserves the local neighborhood relationships of the data points. So the resulting plot you see shows clusters where similar faces are grouped together. And then further we have TSNE, T distributed stochastic neighbor embedding. And after LLE we apply TSNE, and it's a non-linear dimensionality reduction method that focuses on preserving the local structure of the data. So this is mostly good for visualizing high dimensional data in 2D or 3D. Uh, this plot shows the clusters more effectively. And then further we have the UMAP. UMAP is again computationally efficient alternative to TSNE, and it's mostly designed to preserve both local and global structures in the data. So the 2D visualization you see here shows the dataset structure with different clusters. Dataset structure is shown here, and then we have isomap for isometric mapping. It focuses mostly on like preserving the global structure of the data by using geodesic distances. So as you can see, it helps us to see how the data points are related globally in a lower dimensional space. This plot here shows the ability to basically separate clusters. And then we have randomized PCA. So a randomized PCA is a faster variation of basically standard PCA, and then it basically what it does is it approximates principal components efficiently for larger data sets. So the resulting plot here you can see it shows projected the resulting plot basically shows the data set projected onto its first two principal components, so principal component 1 and 2. And then we have kernel PCA. So kernel PCA extends PCA to handle nonlinear relationships by applying a kernel function. So it applies a kernel function, and then which allows us to basically capture complex patterns in the data. So the plot here you see it visualizes the embeddings produced by kernel PCA. And then we have incremental PCA. So for incremental PCA, basically what it does, it reprocesses data in chunks, making it suitable for large data sets that cannot fit into memory. The 2D visualization you see here shows the transformed data similar to standard PCA. And then we apply autoencoders. So what it's basically a neural network based approach, and it allows us to like learn a compressed 2D representation of the dataset. So the encoder, what it does is it maps the input to a lower dimensional space, and the resulting plot shows learned embeddings. That's about it for the first collab, and we'll go on to the next one. So again we are installing all the kinds of import libraries. We are importing and setting up the libraries here, and then we are using a iris data set that's basically for different kinds of like flower iris flowers across three species, and it basically gives this is the structure of the data. So that's sepal length, sepal width, and then uh, petal length and petal width, and then target. This where target is basically the prediction, and then we are applying dimensionality reduction here. Same again, the LLE is done. So this data was a bit different from the previous one. What happened here is it shows the local neighborhood relationships more, and it wasn't like very spread through. As you can see, multiple components are even for TSNE. It's kind of the same, and it's mostly the data set. Even though we applied UMAP, so when we applied UMAP, um, the 2D visualization here basically shows similar results to TSNE. Clearly separating the three species of iris, but at the same time, it's like expected that it's going to be similar on the similar wavelengths for each species, or similar like the clusters will be closer for the same species. And then we had isomap, and for isomap it focuses on the preserving the global geometry of data using geodesic distances. So the results here basically show how this technique captures global structures with distinct class separations. And then we have randomized PCA. So for the randomized PCA, we use it to speed up computations, and it's faster than the normal PCA. And the visualization here shows a clear separation between three classes, and it is using first two principal components of those classes. And then we have the kernel PCA. This is also a nonlinear. This is also a nonlinear transformation that applies a kernel function. So the 2D plot you see here reveals a nonlinear relationships. Reveals all the nonlinear relationships in the data, and then it provides better separability than the standard PCA. Then we have incremental PCA. So for incremental PCA, processes the data in smaller chunks as discussed before. And for the iris dataset that we had, the results are nearly identical to randomized PCA. Right here, randomized PCA, it's pretty identical to the randomized PCA, demonstrating this efficiency and scalability of incremental PCA. And then we have factor analysis. Last time we had encoders, so finally applied a factor analysis which focuses on identifying latent variables. So the 2D visualization we can see here highlights the underlying structure of the data, especially separating its classes based on latent factors. That's it. That's about it for this one, and then we'll jump on to the next one, which is for data bricks. And so we have this data set we use for wine quality. It's a data set from the UCI database, and initially the setting up and everything was a bit difficult with Databricks, but we went on to do it. So we applied dimensionality reduction techniques here on the wine quality data set. To be precise, it's a red wine quality, and then we'll just jump in directly to the code. So here we have the data set selection and loading. So and then we reviewed the data, first few columns of the data, or the head of the data, first few rows of the data, or the head of the data, and then here we are pre-processing it and applying PCA. So somewhere around we are extracting features and then standardizing them, and then we apply PCA right here. Then we get the PCA plot here, and it's pretty much similar to what all the steps we did before. It's just that we are demonstrating it using Databricks. Right here we did we used TSNE. So this is specifically designed for high dimensional data visualization, as mentioned before. So we need a perplexity of 30 and a learning rate of 200 to achieve an optimal layout. So by default, I think it takes 200, and we have a visualization right here for TSNE. It clearly highlights distinct clusters in the data. As you can see, the yellow ones, green ones, and then the dark or black ones. These are the differences, and it separates them. So on the right side, it's more dark. Left hand side, it's more of the light ones. And then we have further installed UMAP and applied UMAP here. So the UMAP visualization plot we have here, it demonstrates well separated clusters. So the separation is good between, as you can see, there's a good spacing here. So well separated clusters have been shown here that align closely with the wine quality labels, and then it is computationally efficient and great for exploratory data analysis. And then right here we have isomap. So for isomap, it's mostly used for datasets with non-linear relationships, as I've already mentioned during the first two collabs. And what this plot shows is it captures the structure of the dataset, though the clusters are not as distinctly separated as with the previous dimensionality reduction techniques. This one is more like more joint or more close. The clusters aren't that separated, so it's not easy to visualize this. And then we further applied LLE, so locally linear embedding. So this focused on preserving local relationships between the data points in the lower dimensional space. So it's more towards the side of the data points that were closer. We like sampled them and then visualize them using LLE, and then this is how the plot looks. The basically it doesn't separate the wine qualities as effectively as other methods, since it's mostly a local focus. The all the clusters are pretty close, and that's about it for this technique and for this collab. Thank you so much for watching. Bye.